So, good afternoon, everyone. And as you see, there's no fire, luckily. However, I'm viewing this as my audition for a chat show host session for this afternoon. I think it's the first and last time that I'll actually be doing something like that. But anyway, it's good to have a change in the format. And this is a slightly different topic than we've listened to so far. So I guess that's why the organisers agreed. I came up with the idea to do it this way. So we're both very happy with it. And we'll start by introducing ourselves. So Kelly, would you like to go first?
Sure, thanks Maggie. So hi everyone, pleasure to be here. My name is Kelly Nichols. I am a recent addition to the Department of Animal Science here at UC Davis. I'm a dairy nutritionist. Before I joined the department last year, I lived for the last 10 years in the Netherlands. I did my PhD at Wageningen University and then worked for the last 5 years actually in industry research for Trow Nutrition in their ruminant research team. So I moved here last year and started the position here in the department. My research I've been grateful to always be able to work on the same topic, which is protein nutrition, actually, in ruminants, mammary gland metabolism, amino acid metabolism. So I appreciate the organizers giving a 30-minute free time to someone who actually doesn't work on methane. Let's hope we don't go too rogue and just talk about nitrogen the whole time. But that's actually my background. Maybe that is why I was asked to speak here, to give maybe some different perspective on this question about the other aspects of physiology that we need to consider when we talk about methane mitigation. So, Maggie, also very happy, I have to say, to share the stage with someone who has had such a long career in this space. I'm honored to be sitting up here with you.
Thanks very much, Kelly. And my name is Maggie Gill, and the last time actually that I was on the Davis campus was working with Professor Lee Baldwin and Professor Eric Bradford in the 90s when we were doing a report together which was called the Council of Agricultural Science and Technology report about livestock. And I was so pleased earlier, I was talking to someone who'd actually was aware of that report because it was a long time ago, but it was a very seminal report in one thing it came up with. But just to give a little background of myself, I was a ruminant nutritionist a long time ago. I moved on then from that more to program management and advice for governments on agriculture and environment more generally, and food systems. And I've done that both in the developing world with low and middle income countries, working for the UK government's aid programme, and also working for both the UK and indeed the Scottish government. But going back to the report that I mentioned, the CAST report, and Jim Altgen, who's here, was someone else who worked on that report. And it came up with a very interesting concept, which was the one of reporting efficiency in terms of human edible or human inedible various nutrients, so protein or human edible energy, in fact. And the key thing about that, in terms of, and it links back to the communication session that we had before, because it's a way of communicating that actually we heard about the world without livestock. I've worked on food security, and I'm very well aware that we could not feed the world if we didn't have livestock doing the sorts of roles that that documentary is actually mentioning. And that's because the rumen, and we've learned a lot about the rumen, heard a lot about it, and the negatives of it, producing methane, but it's the rumen and the microbes in the rumen that actually turn that fibre, the grass, into something which is human edible. And that was the point that Lee Baldwin and Eric Bradford came up with. So it's great that we're here in Davis to actually be discussing that work. So my career's mainly been in on grass, feeding ruminants grass, silage, etc. I was very taken by Rod Mackey's slide yesterday. Don't know how many of you were here for that. I'm not entirely sure I agree with the concept he was using, Waterloo, being from the UK. That is a bit of a sort of negative, but the point he was making, there's an awful lot of data on total mixed rations in relation to methane. There's a lot less in terms of, and the strategies coming up, as we've heard in the last 2 days, don't really work as well when we're thinking about high forage diets or the low quality diets that are used a lot in so many other countries. So another thing we've heard in the last few days is variability. Variability in the results that are out there. And how do we understand those? So Kelly and I came up with this diagram that you see on the screen behind us to show that actually, while there's been so much focus on the rumen and its microbes, they're not the be-all and end-all. And actually, it's the animal as a whole that is the driver in relation to this. So we've heard many analogies during the last few days as well. I want to add another one. Some of you might have spotted that under brain, I have conductor. And what I would like to sort of put across is the fact that it's the brain that is getting the score. If you think of the brain as a conductor of an orchestra, all these other organs that you see on that slide are the different sections, the violin section, the wind section, etc. And the brain is coordinating those sections to deliver the productivity that we've also heard about. And different scores, to me, or different diets represent different scores. They're giving both the brain and all those organs, they're giving them the music to play. They are very different. And our point overall is that these 3 strategies that we've heard of for decreasing methane, so the feed additives, the genetics, and the vaccines, they're all doing the same thing of decreasing the methane, attacking those methanogens, but they're also doing a lot of other things. We don't know exactly what they're doing, and what they do will be different between different diets. So that's the key point to remember, is that the different strategies are not just decreasing methane, and we are going to hear the different interactions. Kelly's going to tell us some results from her research that should be applied or thought about in relation to how these different strategies actually work. And hopefully from that, if we understand more about that, we can understand the variability. And if we understand the variability, then hopefully we can lead to suggest some tweaks to strategies or diets that would lead to producers having the opportunity to actually get the win-win of decreased methane, increased production, or also new strategies that will really address the high fiber diets. So that's a bit too much of me on a conversation bit, but I just wanted to make that clear at the start, and Kelly's going to be doing most of the talking from now on with a bit of sort of prompting from time to time. And we're going to come back to the audience at the end, because between the 2 of us, there's no way that we can answer questions on all of these different aspects, these different organs, how they function. And so if we get too difficult a question, we're going to be sending it back to the audience and see what knowledge is also out there from that. So Kelly, you've worked a lot in nitrogen. Would you like to kick us off by talking about some of the things you've learned from that we have discussed between us are relevant to methane?
Oof, that's an opening. So something we discussed when we were thinking about how to kind of put this together and bring maybe some new perspectives. You, Maggie, mentioned that you had read a recent publication from my team at Trow that was about this idea of post-ruminal nitrogen supply, or specifically post-ruminal urea supply. And I started to think about how that connects to what we're trying to talk about here, other aspects of physiology that we need to consider around the methane question. So what we were doing with that work was basically we spend a lot of time as ruminant nutritionists historically trying to balance the supply of nitrogen and energy in the rumen, right? We try to achieve that synchrony, and we think that that's the key. Myself and my colleagues, and actually we can't take all the credit for that, this idea had been already published in the 50s and 60s, particularly in animals managed extensively, so high fiber, low crude protein diets. This idea that what if we think about enhancing that innate mechanism of urea recycling in ruminants by basically putting the nitrogen, kind of the tagline that I use when I talk about this is nitrogen for the rumen, not in the rumen. So by putting urea post-ruminally and allowing the animal to take a hold of that and deliver it continuously to the rumen as needed. So allowing the animal to take control of its own ruminal nitrogen supply instead of us having, I would argue sometimes, this false sense of control over the nitrogen supply through the diet. So I sometimes say I started my career pretending that the rumen didn't exist. Most of my work has been on post-absorptive metabolism, a lot of post-ruminal infusion models. When I started my role at Trow, we were working on this project. So I had to kind of relearn the importance of the rumen in our ruminants. But I think with that, I've had the opportunity to put a bit of a different perspective on the role that the rumen and the microbes are playing and how, as nutritionists, we're trying to manipulate that. So, that perspective on nitrogen, I think it just brings forward that question of what other ways can we flip around the idea of supplying nutrients, or potentially, to bring it back to this conversation, manipulating methanogens or other microbes in the rumen to achieve this reduction in methane production that we just haven't even considered yet.
Great. And one of the things that as you mentioned, that the nitrogen is then used for is the synthesis of microbial protein. So that brings us back to the different microbes that we've been talking about during the last few days. So do you think there's a potential that changes in that microbial protein are likely to affect productivity if there's different ratios of microbial protein entering the duodenum? So another thing, which there's different strategies for attacking the methanogens, there could also be an impact on the nutrients that arrive in the duodenum, and could that affect production?
Yeah, absolutely. I really liked, towards the end of the day yesterday, we started to get a little bit closer to this idea of nutritional physiology and where that comes into play when we think about reducing, wiping out, using vaccines, whatever it is, inhibiting methanogens in the rumen. I think someone raised the question, what would fill that void if we didn't have the archaea in the rumen? How would the population adapt? From my perspective, again, someone who works on protein, most people in this room are well aware that microbial protein makes up the majority of the digestible protein supply to a ruminant. So there's so many questions you could ask there, but one of them would be, what is the amino acid supply that comes from those methanogens? And just simply thinking we get rid of them, we solve the methane problem, everything is fine. Maybe the next State of the Science Summit will be about nitrogen because now we have a problem over there, right? So what is the amino acid profile of those methanogens, the relative proportion that they're delivering to the small intestine for digestion, and then all the subsequent effects on how the other microbes that are potentially more abundant, but it's an ecosystem, right? So removing those, their capability to grow and thrive, what else is going to be there? How is that impacting this massively important nutritional supply to the animal?
So when I was at your stage of the career, then there were methods around, and we used to measure microbial protein synthesis rates, but there don't seem to be many people measuring that anymore? Have you any comments on how difficult that is to actually measure?
Yeah, it's super difficult. Something else that's been coming up, at least yesterday when I was here listening, is this idea that we have techniques and technologies now that we didn't have at the start of your career, for example. And that's in every space, opening up so many different ways of looking at this problem. So some of the genomic tools, the proteomics, all the things that there's folks in this room who are far more expert in that than me, and those would be the collaborators on the projects then that I'm looking to run in this space. I think that fits very well, getting back to my first point of what are the ways that we've just never looked at this before. Of course, I come at it from the protein perspective, but as I've said, I think we can align those, the methane problem, the protein problem, the nitrogen efficiency problem all together. And what would it be like if we could imagine a situation where we're actually perhaps solving or contributing to a solution to both of those concerns? Maybe that's where we do get that productivity response. If you're able to manipulate microbes with nutritionally by changing perhaps amino acid profile that's going into the rumen, if that's inhibiting methanogens, but also allowing some efficiency in terms of microbial protein synthesis, that sounds like a pretty big win-win to me, and that may actually be a production response that you can measure if we're talking about manipulating metabolizable protein. So it's difficult to measure, but the tools that we have available now, I think, completely unlock a different way of looking at microbial metabolism, particularly from the nitrogen side, and then being able to pair that with all the expertise that we have and are continuing to grow around methanogens and their particular metabolism. They all have their own nutritional needs within that rumen. And I think it's just now that we're able to start asking those more complex questions where we compare those things together.
Right, so one of the things that the arrival of nutrients in the duodenum actually does, alongside the absorption of volatile fatty acids, for example, across the rumen wall, is then to stimulate hormone responses. So you see on the slide that we've put all these pathways in which changes of what's happening in the gut have an impact on the brain. And another one that's been referred to, another physiological mechanism that's had some reference to it during this meeting, is saliva. And anything you'd like to say about saliva and how that plays into this overall response of the animal to different diets?
Yeah, there's a lot we could say about that. Again, kind of from coming at it from the nitrogen perspective, but it certainly applies. We talk about urea being recycled through saliva, but of course a lot of other metabolites and potentially a lot of things that we don't even know about yet. We also know very well that the type of diet that's fed to the animal will impact the relative volume of saliva that ultimately then ends up back in the rumen. So you think of these delivery mechanisms that we're talking about, whether it's vaccines or other inhibitor type products, if we're considering relying on saliva, that might be a question, and again, builds in a bit of that variability between animals that are on more intensive managed diets, higher carbohydrate, or higher concentrate, higher starch, versus our extensive ruminants who are grazing high fiber grasses. The difference in the amount of saliva that's being produced by those diets, that's been well characterized. So that's something that we'll have to think about with any kind of delivery mechanism that relies on saliva.
Right, so the message we're coming across with here is that the animal is responding, the hormones, the neural pathways that are referred to there as well. There's a lot known about these in terms of the human. People who followed human physiology, and if you've got an interest in reading about health, there's been a lot about that gut-brain matrix in terms of human health. So there's a lot that's known there. Obviously, a lot less that's known in terms of animals and livestock in particular, but it's that concept that both the absorption of nutrients are affecting the brain, but the brain will also be sending those signals back to the rumen, to the motility of the rumen? Anything on rite of passage that we need to be looking at as well?
Probably a lot of things. Yeah, I think if we think about where we were 10, 15 years ago in the human space around this gut-brain axis, perhaps at that time people would have said, this is insane that this could ever be connected, or that this will ever be something that we base our diagnoses on, base our interventions on. But that's perhaps the space that we're in now in this area. It's just food for thought, like we have on the slide here. When we're talking about manipulating rumen microbes, we can think about the impact nutritionally, we can think about the impact throughout the digestive tract, but there's other things that's being controlled by other things, the brain, the liver, the kidneys. There's so much other signaling, to get back what you said about hormones. There's just so much more characterization that could be brought to the table, and who knows how that intertwines with all of these interventions that we've been focusing so much on, certainly not to detriment of anything. Like, it's amazing the progress that we've made, but I think one of the through points in all of the panels that I listened to yesterday this is where we're at. We've made great strides. Look at all of the questions that we still have. And I think those are super important to the sustainability of this ambition that we all have. And it's very exciting. It's good that we can't sit up here and say, here, check, 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 we've done all the things. But hopefully this stimulates a little bit of ideas around what are we missing and where can we go based on the new tools we have, these new visions, coming up with some of these crazy ideas like gut-brain axis that years ago, at least in the human space, was very novel, and now it's fairly commonplace. Where could we be in 5, 10 years around that in the ruminant nutrition world, for example?
So we wanted to open it up to the audience to see if anyone had any comments or any questions, although, as I say, this is such a broad area that if we can't answer the questions, we'll be batting them back. We're sure there's someone else in the audience that might be able to answer them. So, hand up already? Yep, sorry. And anyone that's skeptical about this, can we solve the methane problem just by the strategies we have? Do we need to go down this area of physiology?
Hi. Okay. Yeah, that's working. Hi. Thank you so much so far. It's been great. I'm a gut-brain axis person by trade, so I was just curious. Obviously, a lot of the focus in the human world has been around like mental health. Obviously, that's perhaps not so important in the context of cattle, but I was hoping to pick your brain a bit about where you actually see some of these concepts like gut-brain interactions coming into play when it comes to ruminant nutrition.
Anyone in the room? It's very easy for us to stand up here and say, hey, what about this thing that no one's really looked at? But I think it's people who do work kind of cross space. So you say that by training you have gut-brain axis experience. Where do you see it coming into play? The mental health side, but I think in humans it's also evolving past that, so it's that and. So I would say, what do you think? We're certainly not experts in that. I don't know about you, Maggie, but
we're not experts in that area, but I think dry matter intake, I don't know, as a human physiologist, then perhaps you could comment in terms of mental health and intake, which seems to me that actually there is increasing, as the Science Oversight Committee, sorry, I don't think I mentioned at the start, but it's in the bio. I am a member of the Science Oversight Committee of the Global Methane Hub. And within that, we've recently commissioned some work on physiology to do some mapping or find out what's out there about physiology and what the interactions could be. And one thing coming out from that is differences in intake. If you get differences in pattern of intake, or you can get differences in total intake. And I think we've seen from data presented earlier the importance of the methane per kilo of intake, dry matter intake of fiber. So therefore, that potential for the gut-brain axis to actually have an impact on intake will, in the end, have an impact also on the methane production. So, yeah, great question.
If you think about the hormonal regulation too, like all of that feeds back to regulation from the brain, has an impact on intake, has an impact on diurnal patterns. So it could more be a way of just flipping how we're looking at this and coming at it from a different angle, informed perhaps by the human space, and just unlocking some different ways of looking at it.
Yeah, the satiety angle is definitely a great point in terms of intake. I definitely know in terms of like obesity research with the gut microbiome, there has been a lot of study of like how some of the molecules secreted by the microbes affect satiety and food intake in general. So that would be a really interesting application if you were to see, okay, how does this impact, feed preference towards one macronutrient or another, or how does this impact overall feed intake? Yeah, that's a great point.
And we talked a lot about variability between, I loved when Jed said yesterday, no two ruminants are the same. And then we also pair that with the discussion on measuring methane and the concerns around variability. If we can just simply understand more about the physiology of, I know we don't feed individual animals, at least not right now on most dairies, but understanding more about where the variability comes from could help with the measurement, it could help with the interpretation, and it just simply helps the scientists learn more and understand more, right?
Other questions, comments, skepticism? Well, it's the after lunch period for everyone sort of taking up. Yes, we have a hand up.
Can I go first? I'm right over here. Woo.
Oh, sorry, hands in that
No, very
direction.
great.
Sorry.
Excellent conversation. I think that you're tapping into something that there is research in the livestock world. So just recently, UC Davis Extension maybe, and they're here, and they might be able to correct me here, put out a study about livestock on the range that are in predation areas, so areas where wolves have been reintroduced. They found increased levels of cortisol in the hair pulled from animals that were in the territory. So it wasn't even a direct interaction with the wolf itself per se, but just being around a predator. We've also have a colleague who's done studies with aphid behavior with ladybugs. The aphids do not feed if there is a ladybug scent in present. So when you're talking about dry matter intake, I think you're tapping into something that has to do with cortisol levels that could definitely be monitored, and I just wanted to encourage that vein of thought, that there is some supportive research out there, and I think you're in a really cool area. So well done. Thank you.
Thank you for that. It's good to know that, and I'm sure there's huge amounts of data that we could tap into, and as Kelly has said, there's new technologies out there, not just for doing the measurements, but also things like AI or machine learning to actually pull together a lot of that information. Was another question over there?
Yep, that's me. My question is, so given that there could or could not be, I don't know, depending on who you are, this interplay between these different pathways, what is the minimum sample set that we need to be sampling routinely in order to capture these potential interactions?
Tricky questions in terms of that. And Kelly's trying to pass it back. One of the things that in the Science Oversight Committee that we talk a lot about is that with the genetics program, then we have a lot of animals. And I'm sure my colleague, Rob, he keeps reminding us, these are an experimental tool, if you like, that we have a lot of animals that we could take measurements from. So the numbers, potentially getting access to numbers of animals with low and high methane is not so much a problem. I think there are so many different things that you could actually measure. I think our first challenge is what would be the priorities, because those measurements are going to cost money. And I think we need to have a bit more thought, a bit more discussion, a bit more feedback, knowing what other people have already measured, like the cortisol, etc., to actually help us prioritize what it is we could actually measure in some of these populations that are being generated around the world. So I can't give you a specific answer on the numbers, but there are numbers available. It's about priorities, I think.
I think we have time for one more question. Oh, I see one.
It's a race. Who gets to deliver the microphone first?
I've never seen Charles move that fast. Sorry, it's more a comment, Maggie, on your last comment. As having spent a fair chunk of my postdoctoral years working in the neuroendocrine regulation of hunger and satiety, I used to say that it reminded me of the sign over the gates to hell in Dante's Inferno, was abandon all hope, ye who enter here. We have dozens of hormones that are pulsatile released that have been associated with hunger, satiety, intake regulation, and they're the ones we know about. We haven't even touched on the chemicals that are produced by the gut microbiome, which of course, if no rumen is the same, no large intestine is the same. So this is an extraordinary difficult area. So I don't have an answer to your last question as to what do we prioritize here. It's a difficult one, but a fascinating area to study.
Yeah, for sure. Again, the techniques we have now, I'm not saying it's going to be easy whatsoever, and I don't even know if intake regulation is a way to go or the way to go. But I do think it highlights a really unique thing and just hopefully leaves everyone with something else to think about, not even necessarily the intake thing, but just can we look at this problem instead of just putting something in to inhibit? Are there mechanisms inherent there that we can tap into that maybe we never could before? A throwback to my comment about the urea recycling. And then is there, these techniques that we have that we could never look at in that way before? I guess that's the main thing we want to leave you guys with is some of these, we've heard so much over the last 2 days, a lot of kind of state of where we are now, a lot of questions about where to go next, and I guess encouraging everyone to consider the physiology of the animal, what happens when we start to manipulate these really fundamental pathways in the rumen that we might be able to do now, but what happens in a year, 2 years, 3 years down the road to the reality of those animals? Is there still potential on the table to have a production response possible? And how do we use the new techniques, the new knowledge that we have to be able to potentially get there?
Great. So, yes, an excellent question to end on because it should give hope to all the young people in the audience as to just how much of a challenge and therefore how much research still needs to be done. And when I lecture to students, I say, I'm actually quite jealous of all the technologies that you have now to answer some of these questions. So if you're working in this area, a great career ahead of you and a great career ahead of Kelly. So let's join in thanking Kelly for the information she shared.
Thank you, Maggie. Also to Maggie.